we have published about 70 videos on different topics under zoology, and they are all in YouTube channel, in my YouTube channel. So if you have time and interest, try to go through. You will have a good idea about the definition of different chapters, particularly genetics and also embryology, and one or two physiological chapters also discussed. Now here in this video, we give explanation for an important topic under genetics: linkage, crossing over and recombination. We can see the definition and the relevant explanation for these terminologies. Now the linkage may be explained based on the position of the genes on a chromosome. Now here you see the illustration. There are two chromosomes which are said to be homologous chromosomes. Why we say homologous chromosome? They carry similar type of genes, and also the number of genes and the characters they determine all are similar. So these two chromosomes may be called homologous chromosomes. In the same way, for an example, in human being there are 22 pairs of homologous chromosomes, and this XY 23rd pair usually called are not homologous, because X chromosome is longer in size and also different in shape and number of chromosomes compared to that of Y, so they cannot be called homologous chromosomes. Anyway, the genes which are on one chromosome and because of being on the same line, they are said to be linked genes, and the relationship among these genes on one chromosome said to be linkage. Now here you see this chromosome having these two genes, capital A, B, and being homologous, here we mean it as small a, small b. Whether they are dominant or recessive form, it doesn't work. Only their similarity in determining a character, standing for a character. And accordingly here, whichever the genes on one chromosome said to be linked genes, and the process phenomenon of being on a single chromosome said to be linkage. That is the relationship between these genes on one chromosome. Now here, during the meiotic division, that you may be knowing gametogenesis, during gamete formation, there will be the division of primordial germ cells into gametes, and that process of cell division said to be gametogenesis, meiosis. Now during meiosis, there possibility for the exchange of segments between the homologous chromosomes, and that may occur between the two genes. Usually it may occur between two genes. Now here you say capital A, capital B, two genes, and the homologous pair will have on the same line, on the same parallel line, small a, small b. Here there occurs a break between capital A, capital B, and at the same point on the chromosome of small a, small b also possibility for the occurrence of a break. And from the point of breakage with capital A, this segment, the end of this point, will be joining the point of breakage with small b. And similarly, the point of breakage with small a will be joining with the point of breakage with the capital P. Here you see this int mark that indicates the breakage between capital A, capital B, small b, and then the breakage points will be joining the opposite point of the homologous pair. That process of the breakage and this orientation of capital A towards small b and small a with the capital B said to be crossing over. And after this orientation, what happens? The end of the A, that is the end of breakage with small a will be joining the point of breakage at capital B. Similarly, the point of breakage with the capital A will be joining point of breakage with small b. What is said to be reunion of the broken segments. Here there won't be any loss of the chromosomal parts, even you say basically the nucleotides. Simply they will be joining with each other with the opposite chromosome, opposite in the sense homologous chromosome. And here you see this third diagram, capital A after the reunion will have the B as its linked gene. So here this capital A and the small b are the linked genes after the reunion, after the crossing over and the reunion. Similarly, here small a will have its linkage with capital B, and small a and capital B may be called after linkage as linked genes. And the process of the reunion and becoming into the normal gene with the new genes called a recombination. Normal gene, what I mean, now this small b becomes a normal linked gene with the capital A after the reunion. Similarly, the small a and the capital B become the normal genes, after which they will work normal as that of being on the same chromosome. They will not behave like the genes on the different chromosomes. So here the exchange of the segments between the homologous chromosome, usually which may occur between the genes of the linked group, will be resulting in the formation of recombined chromosome. What is said to be, the process may be called a recombination. So now they are the recombinant genes. Even then they will work normal, similar to that of the usual linked genes. So linkage is the arrangement of genes on a particular chromosome, and at the time of meiosis, particularly anaphase stage, there the possibility for, now here you cannot assume this crossing over may occur between all the genes available on the length of the chromosome. It may occur here and there. So if you assume there are about 100 genes on the length of a chromosome, this crossing over process may occur between some one or two pairs. So that way there the possibility for the recombination. Even you can assume the other way, if both genes are dominant, both genes, what I mean, capital A here also, capital B the other also, capital B, even though there the crossing over occurred and recombination completed, you cannot find out any difference in their expression. But anyway, if they happen to be in dominant and recessive form, and from the gametes getting out of their reunion, possibly may show some difference in the coming individual. So accordingly, you can have an understanding of these terminologies. Linkage, crossing over, the result of which will be the recombination. So please share and subscribe to the channel. And I tell you already about 70 topics have been discussed, and definitely you will be having a good idea about the details of the chapters. Please go through.